Hello everyone. I will present you OMArk. So it's our tool for the quality of assessment of protein coding gene repertoires. And by coding gene repertoire here, I mean the set of coding genes annotated on a given genome sequence predicted to code for protein. And here I will mainly, this tool is mainly designed for eukaryotes in particular, wanted to make this precision. And so these coding gene repertoires, they are available on database as proteomes, where they are used for a lot of analysis in comparative genomics. But they are subject to many quality issues that they can be missing genes, they can be fragmented genes, they can be inclusion of non-coding regions predicted as coding genes, or they can be contamination. And although there's tools to detect some of these issues, the lack of tool to detect all of these at once. And that's what we developed OMArk. So OMArk takes as an input a database that's issued from the OMA orthology database that have orthology information for 2,000 species, and that's used as a reference. And it takes as input also the proteome of interest. First step of OMArk is using the software OMAmer. That's a software for fast K-mer-based mapping of proteins into gene families that are represented in OMA as hierarchical orthologous groups. So we obtain a file where each gene is mapped to a gene family, and then OMArk uses this file to provide a quality assessment. So here is how it works. We have the proteome, and we first define an ancestral lineage. That's defined as a latest latest ancestor clade with at least 5 representative species in the OMA database. And this lineage can be either dynamically selected from the placements done by OMAmer, or it can be done using the tax ID of the species of interest. From this lineage, we have the conserved ancestral genes that are the gene families that are defined at the ancestral lineage level. So we think these genes existed in the common ancestor, and it is still present in at least 80% of the species of this lineage. And this set of conserved genes is used to estimate the completeness of the proteome of interest. So we assume most of these genes should be present in the proteome of interest, in which case we assign them as either a single copy or duplicated. And if they are not there, we assign them as missing. And you may be familiar with this concept because that's the same concept that's used in Busco, except OMArk also don't only consider single copy genes, it can also consider genes that exist in duplicated copies in most extant species. But the actual novelty in OMArk is actually the consistency assessments that use lineage consistent genes. That is, gene families with at least one representative that has at least one representative genes from the lineage of interest. So all genes that are known to exist in this clade. And we use that to compare to the assessed proteome to see if the genes we see are the genes from the correct taxonomic level. So if it is the case, we assign them as consistent taxonomically. If they are not, like if they map to other taxonomic level, they can be either contaminations, and this we detect when we get consistent mapping to other taxon, or it can mean consistent when we can't find any pattern in the inconsistent mapping. Finally, we also report the genes that we don't find any homologs for, that can be or not be true genes. So here is our OMArk output looks. So here I gave the example of a turtle. So we have 10,000 conserved hogs, which we use for completeness assessment. We can see this proteome is mostly complete, but still missing around 10% of the genes. And in terms of consistency, we can see like, even though we have most of these genes that are non-homolog, we still find first 15% of genes for which we don't know homologs, but in this clade of particularly might be mistakes, but also we detect a bit of contamination from a bacteria. The final thing that OMArk says also, it measures what we call structural consistency of genes. So genes can be partial mapping, which means only part of the sequence have homology to these known gene families. So that can be indicative of annotation issue too. And we also detect fragments. So fragments that map to gene families that are much shorter than expected. And we can see here there's still a bit of structural consistency issue. So we first validated OMArk on simulations. So completeness simulation was quite easy to do. We took proteomes in database and we removed randomly sequence from it to see how close OMArk could be to the actual completeness. So here on the x-axis, you have the part of the genomes that we kept. And here's the completeness estimated by OMArk, and you can see the black bar here is what we expect as a result. And although we overestimate a bit the completeness, it's really close. We did it for a lot of species, model species, where we are mostly really close to the truth, but also less represented species where it's a bit different, and particularly when there is, for example, whole genome duplication in the species, we overestimate a lot completeness, mostly because there's multiple copy of many genes, and even removing one doesn't reduce the completeness. We compare the results to Busco, and Busco was slightly less overestimating completeness and less impacted by duplication, but still quite a bit. In terms of consistency, so we did another simulation where we generated a random protein sequence from the amino acid distributions of known protein in the proteome, and we wanted to see if OMArk could actually detect that this sequence didn't exist. And in most of our cases, the part of the proteome that was consistent was really close to the expected, which we see we can actually detect erroneous sequences that are placed. So we did other simulations that I can tell you that I could tell you about in the questions. But for now, I wanted also to show the results we have with OMArk. So we pre-computed OMArk results for a bunch of publicly available proteome from many databases, so around 8,000 different proteomes. You can access the website at this link, but also using this QR code. And that's how the interface looks like. So you can visualize the data for any of your clade of interest, where you could see so the OMArk results plus the compared to the protein number in this proteome. You can select the taxon of interest using this burden, so you can look at any clade, quality of proteome for any clades. You can also look for specific species in the search bar. And of course, you can also submit your own proteomes to be analyzed with OMArk, and then you could compare your results to other quality results for the same clade. So one of the things we noticed in the OMArk data that I wanted to outline here, that's for the vertebrate genomes in the UniProt, we could see there was a strange pattern of one taxonomic division with a lot of fragments, around 20% of fragments. And when we looked into it, it was actually bird genomes. But you could see, even though there's fragment in most of them, it's not the case for all. And actually, the proportion of fragments correlates with the annotation source, and most of the B10K annotated genomes, so that were published really recently, have these fragments. And when investigating the results, we could find evidence that this is due to the annotation process, where existing fragmented genes for the previous zebrafinch proteomes were used to annotate these genomes, and the fragments that were present in these original proteomes were also translated there. Second use case I wanted to show you is one thing for which we can use OMArk is to compare different annotation for the same species. Here I show two different annotations for the bumblebee that were present in ensemble. And in the previous version of Ensemble from July 2022, there were 16,000 proteins in these genomes, but OMArk did detect that a significant portion of them had no homologs and were structurally doubtful. And when we look at the most recent annotations that's been a bit more worked on, there's much less protein coding genes in there, but you can see actually the completeness proportion, the completeness detected by OMArk did not drop really a lot, even though we removed 5,000 genes. And in terms of consistency, it is much better. And that's an example I like because it shows a case for also considering consistency when evaluating public data. So here's the take-home message. So OMArk use evolutionary information to evaluate the quality of protein coding gene annotation on two criteria: what should be there, what should not be there. We hope it will help users to refine their own annotation and compare annotation provided by different tools to choose the best one. And it can be used by any user also to select high-quality data in publicly available proteomes. And that, if you want to use OMArk, it's available on GitHub. You can also install the software directly with pip. There is a public web server I told you about, and there's a paper if you want to know more about what I showed. Now, I want to thank you for your attention. I want to thank my lab for supporting me in this work, and I'll take questions.
So we have plenty of time for questions.
So very interesting talk. So I understand that you can identify also contamination at variance with Busco. And I wonder if you notice a particular genome which is heavily contaminated, and what can we do in these cases?
Thanks. So the question is, what can we do when we detect AV contamination in the genome? And that's actually a hard question because you would want to detect this contamination actually prior to the annotation process. And for this, I will actually heavily encourage you to use tools that are that are designed for that, like Blob Toolkit. That's a really good one to detect contamination beforehand. I'd say if you detect contamination with OMArk, it is possible to single out some of these genes, so the ones that were detected, and remove them for further analysis because this list is provided. And we are thinking of also, there's actually a tool in the GitHub that can use OMArk data to also detect the contigs or the scaffolds in the assemblies that have these genes, and these could be removed also for further analysis. But I'll say it's still trying to salvage flow data, and if it's possible, it's better not to use this kind of proteomes.
Thanks for the talk. If I can follow up on the talk, on the question, what are the main sources of contamination you see?
The main source of contamination we detected was mostly from bacterial genomes. We also detect fungal genomes. We did not detect, surprisingly, human contaminations, and we believe it's because we will need a bit more like a really heavy contamination to detect it. But bacterial and fungal, I'd say, are the most prevalent. And sometimes, actually, some proteomes are contaminated by more than one species. That's something OMArk also tried to report. And yeah, that's mostly bacteria again, in this case.
I have a question which I'll ask from here. And unfortunately, it may have been when I was coughing that you answered this question. But about the B10K genomes, you mentioned that there was fragmented annotations because of a fragmented genes in zebrafinch. When you re-annotated, how exactly were you doing that?
Well, we didn't re-annotate it these genomes, what we saw is there was these fragments, and we compared the fragments detected in these proteomes to the ones in the zebrafinch proteome that was used to annotate, so according to the method provided by B10K, and we could see there was quite a high overlap between these fragments. And what's interesting is the latest annotation of the zebrafinch that's been also redone, I think, in the context of B10K. These fragments are completely gone, and there is no this correlation anymore. That's why we think this is mostly ported annotation from zebrafinch to these bird genomes, and we believe that if the new zebrafinch was used with the same protocol, probably this issue will mostly disappear.
Oh, wonderful. We still have time for another question or two, if there are.
Thank you again.